Okay, very good morning to all. Today's topic we are continuing the bacterial transposable element. In our previous lecture, we get cleared with a definition, a pictorial definition of transposable element, and in that two components are very much important. One is the inverted repeat, and in between that inverted repeat, the transposon is there, which codes for the enzyme transposase, and its presence is essential. Then how it going to insert into the another location, that particular part also explained by me. Then later on we are discussing regarding to the various types of bacterial transposable element, and we know that there are two main categories of bacterial transposable element. One is referred as the insertional sequences, IS, and hence they are also referred as the IS element. Another category include the complex transposon. The complex transposon further divided into the three types: composite transposon, TN3 transposon, and transposable phages. In our previous lecture, we also discussed regarding to the various characteristic features of the insertional sequence. These are the simple one as far as their structure is concerned. They may be present on the chromosome, they may be present on the plasmid. The size is varies from several hundred to several thousand. Okay, then one important feature associated with this particular is element is that they not carry any detectable phenotype. Hence for hybridization experiments or the uh, direct observation of lollipop structure under the electron microscope give information regarding to the presence of is element. Otherwise they encodes nothing, only the transposase enzyme is encoded by this transposon, but we can't detect it from the outside. Hence phenotypically they are inactive. Then also I have explained regarding to the meaning of inverted repeat sequence, which is the characteristic feature of all transposable element, and this is the typical structure of inverted repeat. Means here this is a repeat, and and a same repeat is present in an inverted form. Means here AGTC is present, here the AGTC means it is an inverted repeat of this thing. Means this part is repeated as it is, only thing in inverted direction. So that is the characteristic feature. Now in this lecture we are going to continue the topic, and we are going to focus on a complex transposon. The complex transposon as compared to is element, these are the some sort of additional features are there. Means beside the features of is elements whatever characteristic features are there, these are present there. But along with that structures, also other structures are there. And one of the clear-cut difference between the complex transposon and the is element is that the is element do not carry a detectable phenotype. I already given this particular characteristic. But as far as complex transposons are concerned, they carry the typical phenotypic characteristics. Okay, and they are different with respect to their organization is concerned, their structural details are concerned, and they are further maybe of three types. In case of a bacteria, complex transposon may be a three types. One is the composite transposon, other is the TN3 transposon family, and third is the transposable bacteriophage. Just IS insertion sequence are abbreviated as IS element. Similarly, the complex transposons are denoted by the word T, capital T N. TN is also the abbreviation that means the transposon. The first composite transposon. This is the typical structure of a composite transposon. Okay. What happens in this? It consists of the gene which encode the antibiotic resistance, and it is a sandwich or flanked by two identical is element. Means there are two is elements are present, typical whatever structures we are discussed that are present, and that is element sandwiches the antibiotic resistance gene. Here this is the, we know that the we can detect the presence of antibiotic resistance gene by means of phenotypic characteristics. Suppose whether the penicillin resistant gene is present or not, or streptomycin resistant gene present or not, it is very simple. Suppose if you allow to grow the cell in presence of that respective antibiotic and still cell grows, that indicate that the respective antibiotic resistance gene is present, and so antibiotic resistance structure can be phenotypically we can characterize. So that is present here. Only thing means it is actually the made up of two is element, in which in between them, in between their structure, the antibiotic resistance gene is there. Here it is shown that, for example, this is the is1. The structure of this is1 is exactly similar in previously, but here this is considered as a transposon now. Why? Because it is a part of this particular three component: insertional sequence, then antibiotic resistant gene, and then insertion. And the characteristic feature of the is element is that they possesses the inverted repeat, that is also there. In that way, this is the structure in front of you. This is the structure of a TN9. The TN9 possesses the chloramphenicol resistance gene sandwiched between IS1 and two IS1. So these two IS elements are the identical one. It is not like that the here IS1 is present and here IS2 is present. No, both IS1 IS1s are present, and when the such a structure means IS1 two IS1 element are two insertional sequence element plus antibiotic resistant gene which is a sandwich, this particular structure is nothing but the complex transposon. We are going to collect the characteristic features of these one by one. The first thing is that the IS element in the composite units are the inverted or a direct repeat configuration. Okay, the same applicable only the ends of the inverted repeat. That here the difference is that it is an inverted or direct repeat also. Means as it is present. Okay, that is also possibility. Then second is that the two ends of IS elements are themselves a inverted repeat. The relative orientation of the flanking of IS element of the composite TN does not alter the terminal sequence. That they remain same in the inverted repeat. Okay, whenever they are transferring from transposition from the one location to another location, at that time the inverted sequence are playing an important role, because in very first lecture when I started this bacterial transfer, I say that these inverted repeats are actually the site of recognition for the transposes to recognize, then and then the cuts are made, and henceforth the presence of these inverted repeat in form of IS element is important, and in between them this thing, this particular transposon is present. The ability of transposon is determined by the terminal IS element. Why it is referred as a transposable element? Because it is a transposition ability, and these the transposition ability is conferred by the IS element. It is not antibiotic resistance gene which confers the transposition ability, which is the IS element. Hence, for whatever applicable, whatever we discuss regarding to the IS element, it is applicable here. Then there are just for the getting details of this type of transposable element, the two examples are there. For example, in case of TN5, the TN5 possesses the antibiotic resistant gene kanamycin, while the total base pairs which are associated with this TN5 is the 54,000, means it is a longer as compared to the IS element, because IS element plus antibiotic resistant gene is there. Then the terminal IS element which is present here, it is a IS50, and terminal repeat is inverted. While in case of TN9, the antibiotic resistant gene is a chloramphenicol. The base pairs which are associated making the structure of TN9 is 2638, and IS element which are present here, that is the IS1, which is made up of a 768. And here the direct repeat is there. So these actually the repeats decide the how to cut and get inserted into this. Now, one more important thing is that the most of the transpose from one plasmid to another plasmid. So, for example, these TN5 or TN9 comes in the plasmid. Okay. So now here it comes into the plasmid. Now what is actually there? The IS element is there. Here also IS element is there. IS elements are end with the inverted repeat or the repeat palindromic sequences, and mean in between them the antibiotic resistance is there. And suppose if this plasmid carrying transposable element on denaturation we know that the whenever the double stranded DNA is converted um, into the single stranded DNA, it is also referred as a denaturation. Suppose if we denatured it, then the single stranded plasmid is obtained here. But as the one strand is removed from this particular single strand is not a constant one. They always try to make a bonding with the another complementary sequence. And here we know that there is the complementation between, because here 1 2 3 is present, the in inverted repeat 3 2 1 is present. I already given the meaning inverted repeat in previous case. So this is the present. And so what happens? Automatically this part and this part, both the parts are going to get attached. Hydrogen bonds are get formed. Okay. And in that way, here suppose if it is A is there, it's a complementary T is there. When the two G is there, plus complementary G is there. And in that way, this particular region forms the bonding, and henceforth here this is the remaining plasmid, and here is the it is the antibiotic resistant gene. Now these particular structures are very well seen as far as electron microscope is concerned, and due to its typical feature shape, it is referred as the stem or a lollipop structure. The lollipop, why it is referred as? Because it is very much similar to the lollipop, and henceforth a presence of, suppose if you have collected the plasmid and then denatured the plasmid, converted into the single stranded plasmid, and then with appropriate buffer they analyze under the electron microscope. If we found such kind of a stem structure or such kind of lollipop structure are there, that indicate that the these transposable elements are present. So this is the another way by which we can detect. But as far as these complex transposable elements are concerned, phenotypically also we can detect. For example, if the antibiotic resistant gene is there, that gives the hint that the transposable elements are present. Then another feature is that when the transposon is present in the small plasmid, you know that the plasmids are too much smaller also. Even 2 gene, 3 gene plasmids are also there. It consider. In the very smallest plasmid in which the 2 genes are there, the here it is difficult to say. For example, this is the plasmid, okay, and in that this insertional sequence is get inserted, transposed. Means this is the IS element, then antibiotic resistant gene, and this is the another IS. Means you can say that this is the one transposon, okay, composite transposon, here antibiotic. And suppose in the plasmid another antibiotic resistant is there, we can say that this is also, means although a single transposon is present, but due to the presence of a smaller size, it appears that the more than one transposable elements are present. Here you can say that the IS, this is also a transposable element, and ISB and IS is also a transposable. That is the happens in case of a small plasmid. If the number of genes are more, at that time it is appear as a single one. But whenever it is double, we can say that this A is also a transposable element and B is also a transposable element. That is the behavior of a transposon in a small plasmid. Then next family is the TN3 family. Also keep in mind that as we are going further and further, there is a complex structural complexity is increasing. In IS element, only the transposase enzyme, then the inverted sequence. These are the two components of. Now, when we are discussing regarding to composite transposon, at that time we see that the along with that antibiotic resistant gene is also there. Now, in this next stage, that is in TN3 family, it is consist of the large element as compared to the IS element and the formerly discussed transposon. Okay, and it has a sort of a complicated structure. What are the component of this? It possesses the IS elements are not present here. It has its own inverted repeats are there. For example, I am showing you the TN3 family transposons. Here there is a 38 base pair inverted repeat, means 38 base pairs are there here present, and inverted fashion it is also present, means the structural composition is the same as far as these ends are concerned. Only thing is that they are running in opposite direction, inverted. Then there follows the TNPA gene. Then there is the IRS site or location. Okay. Then there is another gene, that is the TNPR. Then there is the BLA gene. Okay, means this is the one gene, this is another gene, and this is the third gene. Means here the three genes are there. Along with that, several locations are there. Means I have shown this double standard two line structure as a gene, and the single line structure as a uh, site or the location, binding site for this different. But actually, everything is a double standard DNA just for the sake of convenience. Means in between 38 base pair inverted repeat, there is the sandwich of several genes. First, it includes the TNPA. Another is the IRS. This is the not a encode for anything. The TNP encode for the protein, and BLA is also encode for the protein. The TNPA encode for the enzyme, that is the transposase. In former case, insertional sequences are there that encodes the transposes, but here the IS elements are not present. Here, its individual gene is present transpose. Then this is the TNP that is encoded by the transpose. Then another is the TNPR. It codes for the resolves. Okay. It helpful in order to identify the location where to make a cut and where to insert. Means some sort of a smarter mechanism is associated with the TN3 family. And blaging, it is actually the abbreviation of a beta lactamase. LA is taken from the lactamase, beta lactamase gene. Okay, so these antibiotic resistance is also there. Henceforth, it is possible to detect the presence of TN3 family by means of detecting the antibiotic resistance. Okay, then resolvase is present, and it has ability to resolve this particular sequence, that is referred as the internal resolution site. So this is the nothing but this site is also referred as the internal resolution site. So that gives the identity that this is the transposable element. And so in order to make a cut from here to here, and for proper binding, the internal resolution site is important because it recognized by the resolvers, and in that way this is the much more complicated structure is there. Okay, and in this thing, in that way this is going to be detached. For the cutting purpose, transposes are involved. For the recognition, resolvers is there, and for that purpose recognition site is also there, and in that way the intact transposable element get detached and get go in another. Then the last type of a transposable element, as far as the bacterial cell is concerned, it is the transposable bacteriophages. So these are transposable bacteriophages. Actually, we know that whenever the bacteriophage infect with the bacterial cell, there are two fates: lytic cycle or a lysogeny cycle. First consequence that they nullify the effect. They denature, they degrade the entered viral genome. This is the first situation. Another thing is that the virus is responsible to degrade the mm, bacterial chromosome, and then it established. This is referred as the lytic cycle. In that, the cell is going to be lysed. And in the third option is that the rather than inactivating the bacterial chromosome, it get incorporated. Means bacteriophage genome is get incorporated. That is also referred as a prophage. But whenever the several bacteriophages genome is get incorporated, many times they are always placed at the one particular suitable site. So, for example, if you analyze the lambda phase, actually these are the part of our next unit where we are going to discuss much more detail regarding the lytic cycle, lysogeny cycle, lambda phase, T4 phase. But here, with respect to transposable bacteriophage, I am going to explain you that whenever the, for example, lambda bacteriophage is incorporating its genome into the bacterial chromosome, the size of site of incorporation is always a constant. Okay, how that I will going to explain at that time. But what is the problem with the other bacteriophages like mu and D108? They never incorporate at a particular site. They can incorporate any part of a bacterial chromosome. This is the first thing. Once they are get incorporated and get lysosomized, means once they become a part of that bacterial chromosome, still they not keep their position fixed. They are changing their position from here to there, and hence this particular capability is nothing of the transposable element, because transposable element get detached from one side and get associated with another side. Hence you'll find that the viral head or tail protein coding genes are continuously changing their site, and that is not applicable to the all bacteriophages. Only the few bacteriophages like mu and D108, and henceforth a whole bacteriophage is changing, and you can imagine that what are the only in case of composite transposon, only the antibiotic resistance gene is there. In case of TN3 family, we have seen that the antibiotic resistance gene is there, then the TNA is there, the TNP is there, means three genes are there. But here, so many genes are there which encodes for the head, so many genes are there which encode for the tail and related allied part of the back. So that all consist of the single transposable element, and it is going here and there, and in that way they are moving. They also possesses the repeat, because this is already given that this inverted repeats are very much important in order to get it detached, because they indicate that from these reasons the cut should be made, and then it is a transfer. Here the in case of a mu bacteriophage, a 56 base pair repeat is present, then mu can insert at a different non-homologous position in E. coli or gram-negative bacteria. Already I have given that there should not a homology. Okay, at any reason it can get involved. Okay, then mu replicate itself by transposing themselves to the loci on the host chromosome. Whenever the chance comes, it also the multiply. Okay, and it does not have the repeated sequence in its termini. In repeats are there, but these are not always as the terminal region. Okay, these are disturbed, means small part only the head and tail, these are also going to be shifted, means even the terminal region is also going to be shifted here and there. And as this is the larger in size, hence it is referred as the largest transposable element as far as the bacterial cell is concerned, and they are also considered as a giant mutator transposon. Why giant mutator transposon? Because already given that the transposition ability is itself the mutational thing. It is a mutation. We already discussed this particular difference at the start of the lecture, that the transposition is a mutation. Yes, it is mutation because it is the changes the sequence. Hence whenever the bacteriophages are changing its location from here to there, it actually inducing the mutation. Hence it is referred as the giant mutator transposon. Why? Because there is a mutation takes place of and that rate of that mutation is the higher one, hence it is referred as the giant mutator. Why mutator? Because it is inducing the mutation, it is changing the thing. Although the individual functions are not get altered, head, tail, protein or the bacterial cells genes are not get hampered, but still the sequences get changed, and our definition of mutation is that whenever the sequences get changed, it is referred as the mutation. Hence these are referred as the giant mutators. And hence they can incorporate the various sites also, and they can make so many combination as far as the bacterial series is concerned. So that's all regarding to the three important types of complex transposon: composite transposon, TN3, and transposable. That is the sufficient information for today's lecture. And uh, remaining topic, next topic, that is how the transposition takes place at a molecular level, what are the mechanism, what are the theories, what are the practical proofs are there as far as the transposition is concerned, that we are going to discuss in the tomorrow's lecture, next lecture. But as far as these things, three things are there, that's all regarding to the at my end. If you have any question as far as the IS elements are concerned, or as far as complex transposons are concerned, you can ask. Yes? No question? Okay.